Hi everyone, this is Ms. Romani, and in this lesson, we will be learning about carbohydrates. Now, you've probably already heard about carbohydrates, or carbs as they are more often called in everyday life. Maybe you've been talking to someone and they mentioned that they no longer eat carbs. So, then what are these carbs? Basically, carbohydrates are sugars, which, yeah, let's avoid these ones as much as possible. But because of these types of sugars, called the simple sugars, which are the leading cause of obesity, diabetes, and most likely also heart disease in Canada and throughout the world. The word carbohydrates and carbs in general are often maligned in many trendy diets. But carbohydrates are actually pretty important to a healthy diet because they are also the starches and fibers found in fruits, grains, vegetables, and milk products. These fruits, vegetables, and whole grains, all of which are carbs, provide our bodies with most of the vitamins and minerals and all of the dietary fiber that keep our bodies healthy and help us fight disease and even help us look good. So, nutritionally, carbs are an interesting and complicated topic. But unfortunately, we cannot devote much of our time to that in this course. If you are interested, though, I have a video lesson on carbohydrates that I recorded for my health science class that you should check out. But this is biochemistry, so let's focus on that for now. So, chemically, carbohydrates are pretty simple. They are organic molecules, meaning that they have a hydrocarbon backbone. But other than carbons and hydrogens, the only other element in their structure is oxygen. These three elements are mostly, though not always, found in a 1 to 2 to 1 ratio, with one carbon and two hydrogen for every one oxygen. This hydrocarbon right here is glucose. You will become very familiar with glucose throughout this lesson and throughout this course. And glucose, like many similar sugars, have the molecular formula C6H12O6 with that 1 to 2 to 1 ratio I just mentioned. Now, this other sugar here is ribose, another sugar you will learn a little bit more about. And its molecular formula is C5H10O5, another 1 to 2 to 1 ratio. In the molecular formulas for these two sugars, the subscripts represent a multiple of the empirical formula CH2O, which shows very clearly that both glucose and ribose are made up of carbons that have been hydrated with water, which is where the term carbohydrate actually comes from. Pretty neat, huh? Now, I mentioned that carbohydrates are important in our diet because of the vitamins, minerals, and fiber that carbohydrate-rich food usually contain. But the sugar molecules themselves have a very specific function. For all organisms, from bacteria to plants to animals, sugars, and more specifically glucose, is the preferred fuel source. For example, the brain is completely dependent on glucose, except during extreme starvation conditions, for its energy. In animal cells, carbohydrates provide us with immediate energy and the ability to store energy for the short term. At least in humans, carbohydrates cannot be stored in our body cells for more than a day at most, typically. In plants, carbohydrates provide immediate energy and long-term energy storage. They also make up the cell walls of plants and the cell walls of fungi. They provide structural support as well as the exoskeleton of arthropods, so like spiders, lobsters, insects. You remember from last year's lesson on biodiversity. Carbohydrates also have a function on our cell membranes. Mostly they stick out towards the outside of the cell, where they can help cells stick to each other or to other surfaces and provide specific recognition sites as well. So obviously, with such a variety of functions, there must also be a variety of carbohydrate structures. And there are. So let's talk about that. Carbohydrates are broadly classified into two subgroups, simple and complex carbohydrates. The simple carbohydrates include two groups. The first is the monosaccharides, which consist of one sugar molecule. The name implies that because the prefix mono means one and saccharides means sugar in Latin. Now, monosaccharides include sugars like glucose, galactose, and fructose. The other simple carbohydrate are the disaccharides, which consist of two sugar molecules bonded together since di means two. Examples of disaccharides include maltose, lactose, and sucrose. Now let's take a moment to notice a trend in the names of the monosaccharides and the disaccharides, and that is that they all end in the letters OSE. In biochemistry, words ending in O's will mean that it is a sugar. The complex carbohydrates include two groups as well. One of them is the oligosaccharides. The prefix oligo means a few in Greek, which makes sense because oligosaccharides are made up of 3 to up to 10 sugar molecules bonded together and no more than that. And we won't really get into this group of carbohydrates for this course in this lesson, but a common example you may have heard about is a group of sugars called fructans. Fructans are part of a group of fermentable sugars called FODMAPs, which are generally believed to be pretty healthy, but tend to give some people, especially those who suffer from IBS, some digestive issues. In the cell, oligosaccharides are often the types of sugars that are found on the cell membrane and help with adhesion and recognition. So we will talk about them when we talk about the cell membrane later on. And finally, there is the polysaccharides, which are made up of more than 10 sugars bonded together because poly means many. The polysaccharides include starches, cellulose, glycogen, and chitin. So let's start with the monosaccharides. There are a couple of ways by which monosaccharides are categorized. One has to do with the functional group it contains, and the other with the number of carbons in the sugar molecule. Monosaccharides have two different functional groups in their structures: hydroxide groups and carbonyl groups. Aldoses are sugars with the aldehyde form of the carbonyl group, and ketoses are sugars with the ketone form. Now, triose, tetrose, which is not pictured here, pentose, and hexose sugars are categorized because they have 3, 4, 5, or 6 carbons, respectively. And let's take a moment to clarify something, because you may be thinking that these structures are not the sugars that you're used to, because so far you have probably just seen sugars depicted as ring structures. But trust me, these are sugars. A monosaccharide can actually exist as a linear chain like these ones, or as a ring or cyclical molecule like you're used to seeing. But what happens is that in aqueous solutions, that is, in solutions of water, they're usually found in ring forms. And since living things are made mostly of water, that is how they're usually found in living things, and that's how they're usually depicted in biology. We will discuss the cyclical sugars in a moment, though. So let's get back to how we categorize sugars. Keeping in mind what you just learned, here is a molecule of glucose. It's a monosaccharide, but here it is in linear form. Is glucose an aldose sugar or a ketose sugar? Look at its structure. It's an aldose sugar. Now, is it a triose, a tetrose, a pentose, or a hexose? Well, you can count the carbons, but you probably remember that glucose has the molecular formula C6H12O6. So it has 6 carbons, which make it a hexose. So glucose is actually categorized as an aldohexose sugar. And glucose is not the only hexose monosaccharide. We also have galactose and fructose, both with a molecular formula of C6H12O6. Like glucose, galactose is also an aldohexose, but fructose is actually a ketohexose sugar. Glucose, galactose, and fructose are all different monosaccharides with the same molecular formula. So they are called isomers. Isomers are molecules with the same molecular formula but different structural formula. So they have the same number and types of atoms in their formula, but they are arranged in different ways. Now, if we look at glucose and compare it to fructose, the structural difference between glucose and fructose is obvious. So fructose is a structural isomer of glucose and also of galactose, meaning that its atoms are actually bonded together in a different order. More specifically, glucose and galactose are aldose sugars and fructose is a ketose. But what if we compare glucose and galactose then? Can you spot the difference between glucose and galactose at all? It's pretty subtle. The difference is actually just right here. The direction of the hydroxyl group in carbon number 4, and just the direction it faces. The difference makes glucose and galactose something called stereoisomers. Stereoisomers means that their atoms are bonded together in the same order, but they have a different three-dimensional organization around one of their carbons. And that simple difference in the position of a single functional group makes these two sugars functionally different in the cells. This tiny difference causes galactose to be less stable than glucose, and as a result, when in our bodies, the liver rapidly converts it to glucose. Most of the galactose that we absorbed in our food is used to produce energy in cells, but only after it has been converted into glucose by the liver. Now, when these sugars are placed in water and become ring structures, the structural differences between glucose and fructose become even more apparent. Both glucose and galactose form hexagon-shaped rings, while fructose forms a pentagon-shaped ring. Both the ring and the straight chain forms of sugars actually exist in equilibrium with each other, but the equilibrium strongly favors the ring forms, especially in water. For instance, in solution, over 99% of glucose is actually found in ring form as opposed to this straight chain form. So let's take a look at how these ring structures form. Place a monosaccharide with 5 or more carbons in some water and it will form a ring structure. This happens when a hydroxyl group in the chain reacts with a carbonyl group, whether that be an aldehyde or a ketone. And this molecule right here is glucose. And in the case of glucose, the hydroxyl group in carbon 5 is the one that reacts with the aldehyde, which is on carbon 1. So let's watch it again. And this time, pay close attention to what happens to that one hydroxyl group at the end. So when the bond forms, the hydroxyl group attached itself to carbon 1 and ended up below the carbon right here. But this was just by chance. The hydroxyl group could have just as easily ended up above the carbon. And as a matter of fact, that is what happens 50% of the time. We call the glucose molecule with the hydroxyl group below the carbon number 1 an alpha glucose, and the one with the hydroxyl group above carbon number 1 a beta glucose. And these two forms of glucose are isomers of each other. And because in biology, like everywhere else, structure determines function, this simple difference in the location of the hydroxyl group between alpha and beta glucose can make significant differences in the properties of the polymers formed by each type of glucose. As a matter of fact, one of these glucoses forms a type of polymer that we can digest and is one of the most commonly consumed carbohydrates in our diet, and the other one forms a type of polysaccharide that we cannot digest, even though we also consume it in our diet. Okay, so let's say we bond 2 monosaccharides together. How about 2 glucose molecules, for example? First of all, the chemical reaction responsible would be a dehydration synthesis reaction, sometimes called condensation reactions. And in the previous lesson, you learned that these reactions form bonds by removing water, and that functional groups will be involved in the reaction. Okay, so if we place then these 2 glucose molecules next to each other, where could the water molecule be removed in order to form a bond? Right here. When the water is removed, a bond is formed then. And now we have a disaccharide, a sugar molecule made up of 2 sugar monomers bonded together. The new bond formed is called a glycosidic bond, also called a glycosidic linkage sometimes. More specifically, for this disaccharide, this is an alpha 1,4 glycosidic bond. Alpha, because the glucose molecules involved here are alpha glucose, and 1,4 because the bond happens between carbon number 1 of one glucose and carbon number 4 of the other glucose. You see, in some cases, it's important to know which carbons on the two sugar rings are connected by a glycosidic bond. So each carbon in a monosaccharide is given a number from, in this case, 1 to 6, with number 1 being the carbon to which in this case, the aldehyde group was attached. The disaccharide formed by this reaction is actually called maltose. And we can't forget about the water, so let's just add that to the chemical reaction as well and include it with the products. So this is the dehydration synthesis of two glucose molecules to form a disaccharide. In this particular case, the disaccharide is maltose. Water was removed and a bond was formed by that removal. But let's not forget that the opposite reaction is also possible. For example, in our digestive system, we can break down disaccharides into monosaccharides. The reaction is called the hydrolysis reaction, and it breaks the bond by adding water in the exact same place where it was removed when the bond was formed. So let's take a look at how the three monosaccharides we learned about can combine to form the three most common disaccharides. For example, plants make a disaccharide called sucrose by combining glucose and fructose. The sucrose made by plants circulates in the sap, and we can actually isolate it from certain plants to make table sugar. Animals make lactose by bonding galactose and glucose. Lactose is the sugar found in milk, and is what gives milk its sweet taste. And as we just saw, two glucose together make maltose. When starch is digested by the enzyme amylase, that's the enzyme that is found in our saliva, maltose is produced. Maltose is also found in breads and cereals, and it's an ingredient that is used when making beer. Polysaccharides are also built by dehydration synthesis reaction and broken apart using hydrolysis reactions. There are three main types we will focus on in this course: starches, glycogen, and cellulose. All three are long chains of glucose. There's also a fourth polysaccharide, chitin. All of these polysaccharides have different functions. Starch and glycogen are both storage polysaccharides. Starch stores glucose in plants, and glycogen stores glucose in animal cells. Cellulose and chitin are structural polysaccharides, making up both the cell walls of plant cells and the exoskeleton of some animals. So these differences in function must correspond to a difference in structure. So let's take a closer look. Potatoes are full of starch, long chains of linked glucose molecules that are a storage form of fuel for plants. It is how plants store any extra glucose for the future. You see, plants will make glucose through photosynthesis, and then will use some of that glucose right away for their energy needs. But any extra is linked together to be used for the future and is stored in their cells in different parts of the plant as starch. For example, in this potato, that's a perfect example of a place where a plant might store a lot of that extra starch. Plants also store a lot of their starch in their seeds, and this is so that the starch can serve as food for their embryo. Most of our grain crops, like wheat, rice, or corn, are seeds, and they are great sources of starch. In starch, the glucose monomers are in the alpha form, with the hydroxyl group and the carbon sticking down below the ring, and they are connected mostly by alpha 1,4 glycosidic bonds. Starch is actually made up of a mixture of two different polysaccharides, amylose and amylopectin. Both of these are polymers of glucose. Amylose is made up entirely of unbranched chains of glucose connected only by alpha 1,4 glycosidic bonds. But amylopectin is a branched polysaccharide. Although most of its monomers are connected to each other by these same alpha 1 4 glycosidic bonds in a very long chain, every 24 to 30 glucose molecules in that chain, it will branch out with another chain, and this other chain is linked to the original chain by an alpha 1 6 glycosidic bond. So the differences in structure between these two starch molecules, basically the fact that one is branched and the other one isn't, gives them different properties. Amylose forms a structure that is not soluble in water, and it is not easy to digest. Amylopectin, on the other hand, is soluble in water and is easy to digest. And these properties are due to the branching that is found in the molecule. So starch molecules are formed in plant cells so that plant cells can access extra glucose molecules whenever they need energy. But what about animal cells? Can we store glucose that way? And the answer is yes, but not using starch. We use a similar molecule called glycogen. Like starch, glycogen is a polymer of glucose, but unlike starch, it can't store glucose for a long time. For humans, our ability to store glycogen is limited. We store it in our liver and in our muscle cells, but the amount we can store will last us about 1 to 2 hours of intense exercise, or maybe up to 12 to 24 hours of just regular activities. And glycogen is a branched polysaccharide that is even more branched than amylopectin. The branches of glycogen extend out of the main chain every 8 to 10 glucose molecules. And just like starch, the chains are bonded by an alpha 1 4 glycosidic bond, and the branches by alpha 1 6 glycosidic bonds. This branching is important because it is the branching that increases the solubility of glycogen. Furthermore, branching increases the places on the molecule where enzymes can both break down or build glycogen. So the branching increases the rate of glycogen synthesis and the rate of glycogen degradation in the cell. So this is how it works. When we eat carbs, say like a potato that is full of starch, we break it down in our digestive system. Enzymes in our small intestine will use hydrolysis reactions to break the starch down into glucose. The glucose then travels to our cells in our bloodstream, enters our cells, and provides our cells with energy. Now, how the cell gets energy from glucose is a topic that we will cover in detail later on when we learn about cellular respiration. So don't worry about that right now. Now, when we digested that potato, though, we absorbed way more glucose than our bodies needed for immediate energy. Some of that glucose was used for energy right away, but a lot of the extra glucose moves into our muscle and our liver cells to be stored for later. In our liver and muscle cells, dehydration synthesis reactions then form bonds between glucose and form glycogen. Later on in the day, when, say, our blood sugar levels drop and we need more glucose for energy, or after exercise when we need more glucose for energy, a message will be sent to our muscles and our liver cells to promote the breakdown or hydrolysis of glycogen into glucose, which can now be used for energy by our cells. So as you may gather, our ability to store glycogen for later is pretty important. It keeps us from having to consume sugar 24/7 so that we can keep our blood sugar levels even between meals. And for athletes, glycogen provides much needed sugar energy during, say, a race. So if you're interested in learning more about how athletes maximize their glycogen stores before a long race, you can check out the lesson video on carbohydrates that I recorded for my health science class, because I get into that in that lesson. But for now, we need to move on because although energy storage is one important role for polysaccharides, they are also crucial for another purpose, which is providing structure. So let's move on to the structure of polysaccharides, cellulose and chitin. So cellulose is a major component of plant cell walls, which are rigid structures that enclose the cells and help veggies like lettuce and celery have that crunchiness to them. Wood and paper are mostly made out of cellulose as well. Cotton is also made of cellulose. So to give you an idea about the difference in the structure between cellulose and starch, think about what starch both looks like and feels like. You might have some cornstarch in your kitchen so you can get an idea. Or if you don't, flour is mostly starch, so that's another example. So starch is super soft and powdery. Now, to contrast that, feel a piece of paper or a cotton shirt. That's cellulose. Cellulose is strong and not at all powdery, which is why it serves a structural function in plant cells, giving plant cells both strength and shape. So, to understand this difference between starch and cellulose, we need to take a close look at the molecular structure of cellulose. Cellulose is an unbranched polysaccharide. It is made up of chains of glucose linked by beta 1,4 glycosidic bonds. That's right, beta bonds. So now beta glucose makes an appearance. And if you're thinking, hey, I wonder if it is the difference between beta and alpha glucose that gives cellulose such a strong structure, well then bonus points for you, because you would be correct. Because it is made up of beta glucose instead of alpha glucose, chains of cellulose can cluster together to form these parallel bundles that are held together by hydrogen bonds. And it is this hydrogen bonds between chains of glucose and cellulose that gives cellulose its strength and its rigidity. Let's take a closer look in more detail. So let's recall. When glucose goes from a straight chain to a ring structure, it can form either alpha glucose or beta glucose. Now, joining two alpha glucose molecules together with an alpha 1,4 glycosidic bond is pretty simple. If we count the carbons, we can see that the hydroxyl groups of carbon 1 and the adjacent carbon 4 are both below the carbons. They're pointing down. And by pointing in the same direction, a water molecule can actually be removed very easily in order to form the bond between them. The same is not true about joining two beta monomers of glucose. Because if we're trying to make cellulose, we're going to have to join beta glucose to beta glucose. So if you take a look at this structure right here, you can see that the hydroxyl groups in carbon number 1 and in carbon number 4 are not on the same plane. They do not point in the same direction. One of them is pointing up above the carbon, and then one of them is pointing down below the carbon. So the water molecule would not be easily removed because they're not positioned correctly. So one way to do that then would be to just flip the molecule. So if we flipped one of the glucose molecules, then we can get the proper orientation and the water can be removed. And that's exactly what happens. So in a cellulose chain, every other glucose molecule is flipped to allow for the formation of the beta 1,4 glycosidic bonds. Now, this might look a little bit overwhelming, so I'll highlight this part of each molecule. And now you can more clearly see how every other glucose molecule in these chains have been flipped. And so I've put two branches together. So these are two separate branches or chains. And notice that at the end, there's a little part that is in square brackets with a subscript at the bottom. That's one way that we can indicate, hey, this chain continues for, if I put N, if I make N say 50, it would mean that this chain continues for 50 more glucose molecules. So this is just a way to signify that this continues on. So we have these two long chains of beta glucose linked together. And when two long chains of cellulose are placed next to each other, they will be able to hydrogen bond by using their polar hydroxyl groups. And this is what gives cellulose strength and rigidity, and why it is such a strong polysaccharide. The hydrogen bonding, that's what makes the difference. Now, before we move on, I want to clarify one thing. I know what you guys are like, and that you're constantly analyzing and questioning, which I love. So I know that some of you have noticed that the hydrogen bonds don't quite seem to line up perfectly between the adjacent glucose molecules. And that is because this diagram does not really represent glucose in its true ring form. You see, we tend to draw glucose as a perfect hexagon because it makes it easier to visualize, especially for high school biology. But what you will learn if you move on to university biology and organic chemistry classes is that glucose actually forms what is called a chair conformation that looks more like this. So using the chair conformation, which is more accurate for the structure that glucose forms in 3 dimensions in real life, the hydrogen bond between cellulose chains would look more like this, which aligns the hydrogen bonds much better. Now, in this time, I'll highlight the hydrogen bonds for visibility. And so now you can see how extensive the hydrogen bonding is between cellulose chains. And that is what gives cellulose its strength. Now, one more thing about cellulose. The glycosidic bonds in cellulose can't be broken down by the human digestive enzymes. We don't have the enzymes to break down beta glycosidic bonds. So we are not able to digest it. We're not able to digest cellulose. That's not to say that cellulose isn't found in our diets. It is actually an extremely important part of a healthy diet. We call it fiber or dietary fiber. And even though it is just passes through us undigested, it plays a vital role in slowing down our sugar absorption and reducing our risk of diabetes and heart disease, and also some cancers. It also helps to keep us regular and to keep us feeling full when we eat. Now, some herbivores like cows and sheep and buffaloes and horses and koala bears have specialized microbes in their guts that actually help them digest cellulose. So they themselves don't produce the enzymes to digest cellulose either, but they do have this symbiotic relationship with a special microbe or organisms that live within them that will digest cellulose and then will release the glucose in cellulose, and then they'll be able to utilize that glucose. Wood chewing termites also break down cellulose because they have these microorganisms in their guts. So, speaking of termites, insects and all other arthropods make a polysaccharide called chitin to form their exoskeleton. An exoskeleton is a hard external skeleton which protects their soft inner body parts. And if you've ever stepped on an insect and heard a crunch, you know that chitin is pretty strong. So chitin is a sugar. Fungi also produce chitin. While the cell walls of plant cells are made out of cellulose, the cell walls of fungi are actually made out of chitin. So chitin is a very strong sugar, and its structure kind of resembles cellulose, but it is made out of modified glucose units, not just regular glucose like all the other polysaccharides that we've seen so far. And these modified glucose units, called beta N-acetylglucosamine, is basically a beta glucose that has had its OH group in carbon number 2 replaced by this new nitrogen containing functional group that we're not going to get into. Now, chitin is just long unbranched chains of this beta and acetylglucosamine linked together by beta 1,4-glycosidic bonds, just like with cellulose. And just like in cellulose, because chitin is made out of beta glucose chains, chitin can hydrogen bond between those chains. And it is, again, that hydrogen bonding that gives this molecule such strength. So it's all about hydrogen bonding when it comes to strength, at least with sugars. So to summarize, the 4 main polysaccharides are starch, glycogen, cellulose, and chitin. Starch and glycogen are both storage polysaccharides that are used by plants and animals respectively to store glucose. Both are made out of alpha glucose chains. Some are branched and some are unbranched. The ones that are branched are able to dissolve in water. Cellulose and chitin are both strong structural polysaccharides and are both made out of unbranched beta glucose chains. And these beta glucose chains can gain their strength because they can hydrogen bond between them. And that's it for this lesson on carbohydrates. There was a lot of information in this one lesson. So feel free to watch it again if you need to pause it, and Hopefully you didn't watch it all in one sitting and maybe took a break halfway through, but if you didn't, well, good for you. So I will talk to you later, guys. Bye.